Good morning to all of you. Today we will see something about the DNA recombinant technology and its applied aspects, and also the process by which it is being conducted. After completing DNA recombinant technology, we will also see some points about the polymerase chain reaction and also about the various blot techniques. Now, DNA recombination is a process by which the DNA from two different sources are recombined. Now, even within the cell also, during the cell division, there is a DNA recombination taking place, especially during the mitosis, whereby the homologous chromosomes come to lie side by side, and there is an exchange of chromosomal material between these homologous chromosomes. So this is also an example of DNA recombinant process. But in the lab, the DNA recombinant technology pertains to when two DNA molecules coming from two different species or sources are recombined to form new genes. That is also DNA recombinant technology. Now, DNA recombinant technology or the genetic engineering, as it is known nowadays, has got several applications. Now, for the production of a vaccine for hepatitis B virus, it is necessary to pool the hepatitis B virus containing serum, and during these processes there is a chance of the technician or the doctor getting infected. So such infections can be prevented by preparing the vaccine by DNA recombinant technology. Also, we can get human proteins synthesized in the bacteria, for example insulin and growth hormone. Now, another aspect is that we can diagnose various types of genetic diseases like sickle cell hemoglobin or cystic fibrosis or Duchenne muscular dystrophy or hemophilias with the help of both the DNA recombinant technology and also polymerase chain reaction and the Blot techniques. So all these molecular techniques have immense applications. Now, what are the tools required for the DNA recombinant technology? The various tools required include restriction fragment length restriction endonucleases, restriction endonuclease enzymes, which are known as molecular sieves. They can identify specific sequences of 4 nucleotides, for example AATT and TTAA. That means in both the strands the same sequence is present. Both if you read forward and backward, the same sequence will be there. Such sequences are known as palindrome sequences, and there are specific enzymes which can identify these sequences and cleave the molecule at these palindrome sequences. So, restriction endonucleases are many types. There are almost more than 800 types known today. All of them have sequence specific nature, and they cleave the DNA molecule at specific sites only. So we can select what type of restriction endonuclease we want to employ. Now, another tool that is required is the vectors. These vectors are actually carriers of human DNA into the microorganisms, that is Escherichia coli. Now, there are 3 types of vectors. One is known as the plasmid. Plasmids are actually circular DNA molecules present in certain viruses, and plasmids are responsible for certain bacteria, and plasmids are responsible for conferring antibiotic resistance. They multiply autonomously. They are extra chromosomal in nature. Now these plasmids can be cut at specific points with the help of restriction endonucleases, and to this cut site, which may be either producing a sticky end or a blunt end, the human DNA could be incorporated. Now another type of vector is known as the cosmids. Cosmids can take up larger sequences of DNA, whereas plasmids take up relatively lower length DNA molecules. Now, if still larger DNA molecules are to be incorporated, then we should use the bacteriophages. So these are all known as vectors, and then what else is required? Now, first is to get the target DNA, human DNA, which we want to incorporate into the plasmid, and then later on to the bacterial culture. Now, for this, for example, we want to isolate the insulin gene. Now, insulin gene is such a small fragment of the chromosomes. To find out insulin gene and isolate it is next to impossible, because insulin gene is only about 0.0001% of the total human genome. So it is very difficult to isolate the human insulin gene. So what is the alternative? The alternative is to isolate the insulin mRNA. Now, as we know, although the life starts with at the zygote, and every cell of the individual consists of all the genes that are required to synthesize all the proteins. But in certain tissues, only certain proteins are expressed, and many other proteins are not synthesized. Now, for example, in the beta cells of the islets of Langerhans or pancreas, mainly the insulin is synthesized, whereas in the alpha cells of the islets, the glucagon is synthesized. So if we want to isolate the mRNA of insulin, we should better look into the beta cells of the islets of Langerhans. Now, by a technique of RNA extraction, all the RNAs of the beta cell can be extracted, and then this mixture of RNA could be passed through a column which contains polyuracil derivatives. Now, as the column contains poly-U and as the mRNA contains poly-A tail, now all the other RNAs, that is transfer RNA, microsomal RNA, or the ribosomal RNA, all these RNAs are eluted, whereas the messenger RNA will be held back. Now, by a different eluent, it is possible to elute the messenger RNA also. Now, this messenger RNA, from this mixture of messenger RNA, it is possible to isolate the insulin mRNA. Now, this insulin mRNA can be acted upon by a reverse transcriptase, that is RNA dependent DNA polymerase, so as to get a single-stranded DNA molecule. Now, from the single-stranded DNA molecule, it is possible to get a double-stranded DNA molecule, which is now representing the insulin DNA. Now, we have prepared an insulin DNA from the mRNA of insulin. It is a much easier job to do. Now, this insulin gene, which is present now, should be processed by the restriction endonuclease enzyme, and this will be the same restriction endonuclease that cleaves the plasmid also. Now, due to the base complementarity, the insulin mRNA can be incorporated into the plasmid So this is now known as the recombinant plasmid. Now there will be so many plasmids without the human DNA, but there will be at least some plasmids carrying the human DNA. Now this recombinant DNA plasmid of the DNA plasmid along with the other plasmids are incubated with the E.Coli bacteria, and the E.Coli bacteria imbibes all these plasmids into its cell. Now there will be plasmids without recombinant DNA also. But what we are interested is the plasmid carrying the recombinant or the host or the human DNA molecule. Now, how to select which bacterial culture carries the human DNA recombinant plasmid? Now, for this, the response to antibiotics is resorted to. That is, all the wild bacteria, they grow in a natural media and they are not killed. But if we add the antibiotics tetracycline and ampicillin, then it can be seen that only those bacteria which has got a plasmid will be thriving. I mean, both without a human DNA and with a human DNA containing plasmid, and the bacteria will grow. That means the bacteria containing both the plasmid without human DNA and the bacteria which contains a plasmid with recombinant DNA also will survive. Now, if we add further, and all the other bacteria will be killed. That means the plasmids actually confer antibiotic resistance. Now there may be at least 5-6 colonies which are thriving, and out of these 5-6, some of them may be carrying only the plasmid, and some others may be carrying the plasmid with the DNA recombinant fragment carrying the human insulin gene. Now, if we do the replica plating of these survived organisms, then we can see that in the replica plate, one of the colonies is found to be missing. That means, in the original plate, if there were 6 bacterial colonies thriving, but in the replica plate there are only 5 present. Now, where this 6th one has gone? The 6th one, is dead because in the replica plate we are adding chloramphenicol antibiotic. So what happens? The chloramphenicol antibiotic, if it is added, the portion of the plasmid which carries the antibiotic resistance to chloramphenicol is split up due to the incorporation of the human insulin gene, right? So the chloramphenicol antibiotic resistance portion of the plasmid is disrupted, and no longer chloramphenicol is no longer the bacteria is able to counteract the effect of chloramphenicol, and so the bacteria will be dead. Now, actually, it is this dead colony that we are interested, because they contain the human insulin gene inserted into the bacterial plasmid, and this is due to the chloramphenicol resistance portion of the plasmid is split up by the human insulin gene, because it is at this point where the human insulin gene is recombined with the plasmid. So if we look for the corresponding colony in the original plate that represents the dead colony, then we are able to isolate this bacteria which has got the recombinant plasmid present in it. Then now, as the recombinant plasmid carries the human insulin gene, these bacteria can be isolated, and then further experiments can be done with this colony. So that means this insulin gene can be further transcribed into insulin mRNA, and this insulin mRNA can be further translated into the insulin protein, which is the hormone, and this can be used for the treatment purposes, especially mainly for the diabetes mellitus patients. So in this manner we can synthesize the human proteins from the bacterial colony. So this is an application of the DNA recombinant technology. Now another technique that will be interested, is the PCR, that is the polymerase chain reaction. Now the polymerase chain reaction means we can get several copies of the same DNA strand. Suppose we have got only few DNA strands with us, and we want to magnify, or rather increase the number of copies of this DNA strand. Then it is possible to do, because it is always not possible to get enough DNA strands from the individuals or from the place. So for this, what we need are some primers are required, because primer is an RNA primer. RNA primer is initiating the DNA replication or the DNA synthesis. Now the double-stranded DNA is heated at 90 degrees, and hence the two strands are separated, and each of these two strands will serve as a template for the further multiplication of these DNA molecules. So, but we, in addition to the primer, we also need the Taq polymerase, known as the Thermus aquaticus DNA polymerase, because this is derived from the bacterium, that is the Thermus aquaticus. Now this will start polymerizing, and the temperature is brought down from 90 degrees to 50 degrees, so the enzyme can act, and also the substrates of deoxy ATP, deoxy GTP, deoxy CTP and TTP are added into the mixture, and gradually several copies of the DNA could be obtained in this manner. That's why it is known as polymerase chain reaction, or PCR, and this large number of DNA molecule can be used for various experiments. Also, this will help us to find out whether the DNA of a person who has committed a crime is matching with any other suspected individuals. So it has got medical legal applications. And if we take the DNA from a patient with sickle cell anemia, or if we take DNA from a suspected individual of sickle cell anemia, it can be diagnosed whether the person is having sickle cell anemia or not. So this is a very important technique to be performed. Then the presence of a virus or a bacteria in the body also can be detected by PCR. Now there is what is known as we know that people get tuberculosis, and it is very difficult to diagnose in the initial stages, whereas the sputum analysis may not be confirmative. But if the sputum contains a few tubercle bacilli, then it can be identified by a technique of polymerase chain reaction. Now, yet another technique that is of great significance is the blot technique. Now, there are three types of blot technique. One is known as the southern blot, second one is the northern blot, and third one is the western blot. Now, the southern blot technique detects the DNA molecule, whereas the northern blot detects the RNA molecule, and the western blot detects the protein molecule. Now, what we need are probes also. So, from the cell containing the target DNA is extracted, and then the DNA is denatured, and so the DNA strands become single stranded, and then it is electrophoresed. Now, on an agarose gel. Now, there will be several fragments of DNA obtained because we have used first the restriction endonuclease enzyme, which will be cutting the large DNA molecule into small fragments. So, we get so many number of fragments, and they migrate to different levels, and out of these one or two fragments may contain the desired DNA. And for this, we have to use what is known as nitrocellulose paper, which will be blotted onto the agarose gel, so that the complete pattern is blotted onto the nitrocellulose membrane. And then we have to add a complementary probe. Suppose we want to detect the HBS gene, so we have to have a probe for the HBS gene. Or if we want to detect the cystic fibrosis protein gene, then we should have the corresponding probe for this. Now, these probes are available for several abnormal proteins and which can be selected, and in this manner several genetic diseases also can be detected. Now, after pouring the radioactive probe, now probe is actually a single stranded DNA molecule which is complementary, which possesses a complementary sequence to the target DNA. Now, if the target DNA is present in the mixture, then there will be a specific impression produced on the autoradiograph after keeping in the dark for a couple of days. So, the DNA, the abnormal DNA can be detected. We can also detect the oncogenes, the absence of oncosuppressor genes, or the mutated oncosuppressor genes also in this manner. Now, the northern blot technique employs the detection of the RNA, not the DNA. So, here the probe can be either a single stranded DNA or a complementary mRNA. So, mRNA can be detected in this manner. And the third technique is the detection of proteins, and these proteins, the protein of the viruses or the bacteria if there is an infection, or the protein HBS, that is sickle cell hemoglobin, also can be detected. But here we have to use an antibody because they are proteins. So, antibodies are combining with specific proteins. In this manner, these techniques can be performed.